Thank you so much for the invitation, and thank you so much for giving me the opportunity to learn something new. So I really see this lecture as a way to build on Rachel's beautiful lecture. So that was a great introduction to a lot of my work that I will talk about, but also to the stage, we need to take a step back and look at the niche that the T cells reside in and what happens with the whole tissue-wide skin in psoriasis and during resolution. This was a challenging title, so thank you, Curtin, for really putting me on the spot here, because there's not a lot published as of yet. I would have loved to have this lecture in 2 years' time, because I think there will be a lot more then, and it is a bit of an introduction for what is to come after the pause. So stay, because I already saw that there are 2 very interesting epigenetic talks coming on then. And this is just a tower, beautiful building, and the Leo Foundation Skin Immunology Center is up here on the 12th floor. So if you're ever in Copenhagen, come visit us. It's a magic place for research. And these are my disclaimers. So as a clinician, and this is a wonderful study that Mona Ståhle initiated in Stockholm in 2006. Patients were followed in the non-interventional way for 10 years after onset of psoriasis. These patients were recruited from all over, and as you can see, the majority of these patients had a mild disease. They were treated as the need arose, so the number of severe to moderate cases did not really increase. But what I thought was striking when I met 125 of these patients after 10 years was that they were pretty stable. They had an onset with a mild phenotype, and they still had their mild phenotype 10 years later. So as clinicians, we have not done very well with this subset of patients. And these are huge numbers. There is a need for topical treatment or mild treatments for psoriasis that gives long-term resolution, just as Rachel already told you about. So this is a major group. The other thing that was amazing with these patients who just came in out of the goodness of their hearts to talk to us was that 20% had minimal disease activity. So obviously disease resolution and tissue modification and normalization can happen. So I think this is really promising for everything we do. And I'm very interested in this subset of patients to understand their focal homeostasis in the resolved lesions. So when we look at the skin, we have the T cells in blue, we have dendritic cells. This is a plaque, chronic psoriasis, inflammatory dendritic cells in green, and Langerhans cells in red. But the majority of cells are stroma cells, keratinocytes, and fibroblasts. And as you can see, this is really a chronic tissue-wide remodeling of the skin with lymphoid-like structures around the vessels here in the dermis, and then this very thick epidermis with a completely changed differentiation pattern of the keratinocytes. Coming back to the T cells, they do stay for a long time. In resolved lesions, this is a patient who'd been on anti-TNF treatment for 4 years. 1/3 of his CD8 resident T cells responded with IL-17. And here we defined resident T cells by CD103. I'll come back to why we didn't really go into CD49A at that point. But what was striking to me was that the Langerhans cells, these cells, when we look in a microscope, you find microclusters around the vessels, microclusters of highly inflammatory cells in epidermis, always in very close contact with Langerhans cells. And these Langerhans cells, they keep producing IL-23 and IL-15, survival factor for IL-17 TRM cells. So they don't live on their own. They need some local stimulations to survive. Another thing that I found fascinating about psoriasis, these are just primary keratinocytes propagated from individuals with psoriasis or without psoriasis. And when these individuals are exposed to cytokines that we know are upregulated in psoriasis plaques, you can see here that there was a tendency, a slight tendency, that it was more CCL20, and CCL20 is the ligand for CCR6. CCR6 is a chemokine receptor that marks cells prone to IL-17 production. When these keratinocytes were stimulated IL-17, there was a tendency of an increase with more CCL20. This was not very impressive. But then when the cytokines were combined, it was a striking difference in the CCL20 production between healthy keratinocytes and keratinocytes from psoriasis individuals. And this was not the case for many of the other chemokines and cytokines that we measured in these samples. So obviously, T cells in psoriasis are in a sort of pro-inflammatory environment. And when we look at local relapse, it is also due to lower threshold to psoriasis inflammation. And this is one of the very few studies that actually shows that psoriasis comes back in the same spots. We all talk about it, but if you go into literature and try to look for these studies who have been consistently mapping lesions before, during, and after resolution, it's hard to find any. So this was a young medical student and a PhD student in Lars Iversen's lab in Aarhus in Denmark, and he photographed patients before and after climatotherapy and found this striking pattern of local recurrence. So we have local relapse, we have a subset of disease-driving T cells that are retained for years, and we have dendritic cells and keratinocytes that are poised to psoriasiform inflammation. So how does this work? And why is it so long-lasting? A lifespan of a keratinocyte is on 40 to 50 days. In psoriasis, as you know, it's very shortened. And the lifespan of resident T cells remains to be shown. It's very hard to do these experiments. We would need some single-cell carbon-14 really to know for sure, or a single-cell deuterium measurements. But circulating memory T cells have a predicted lifespan of some 200 days or so, and it would surprise me if we had year-long survival of T cells in the skin. And yet, we find these very long-term disease memories. So what happens? What keeps the inflammation? We have talked, and it's really the circle between chronic skin inflammation and resolution, where we have treatment and relapse due to different triggers. We have the cellular composition that I have talked about, and then, of course, all the different levels of inflammatory states on the genome level, the epigenome, transcriptome, proteome, and post-translational modifications. So I will now talk about the publications out there on epigenome and epigenetic changes in psoriasis. So what is epigenetic wiring? Well, it dictates the availability for chromatin for transcription of gene expression. It's a very physical thing. We would never fit all the chromatin and our chromosomes in a cell if it was all unpacked and available for transcription at all times. So it is very closely packed into our chromosomes. We have histone modifications, the DNA spirals around it, and then DNA methylation. This is a lovely figure from Johan's comment on one of the first that I know of epigenetic analysis of psoriasis skin. And you can just see that either with the methylation, if you have methylation, the gene is closed. But when genes are hypomethylated or demethylated, the gene can be actively transcribed. Same thing with histone modifications. You have repressed genes and expressed genes, and this can be measured with a number of different methods. So this is all. It's quite easy, difficult to do though. And the first study that I'm aware of came from the Bocock lab in 2011, and they looked at biopsies from patients with psoriasis, lesional or non-lesional biopsies, and normal skin. And this was a high-throughput genome-wide bead array. Methods have been much improved since then, but there were both increased methylation and decreased methylation in psoriasis versus normal controls. And note here that when we look at the non-lesional psoriasis versus normals, there were very little differences found in this material. But the differences that were found in psoriasis were at large confined to the epidermal differentiation complex. As expected, we only have to look at the histology to understand that there's a lot going on there. And then there were, here you can see in green, the hypomethylated, the open sites, and in red, the hypermethylated. So in psoriasis and in healthy non-lesional psoriasis, you just had a different pattern. And of importance, epigenetic changes seem to be reversible after 1 month of TNF treatment, which was promising and interesting. Sang et al. Published another story in Journal of Autoimmunity a few years later, also looking at DNA methylation, where now there were more differences with both hypo and hypermethylated sites in non-affected psoriasis lesions. Let's see where we are. Here we are. Versus normal skin. More and more sites were starting to turn up. And what they did, which was very neat, was just to look at TIMP2, which is a metalloproteinase inhibitor. Metalloproteinases are upregulated in inflammation. They're very important for tissue remodulation, and TIMPs inhibit them. And you can see here that the methylation of TIMP2 was correlated to the POSIX score and reversely correlated to the mRNA expression. The more methylation you have, the less gene expression you will have. And then Charlotta Enebeck from Linköping in Sweden dug into the non-lesional psoriasis skin in a paper in 2018. And she found that with better techniques, and this was bisulfate sequencing, she found that if you look at the psoriasis, and this is also looking at methylation, you have a clear difference between psoriasis and controls. But the uninvolved psoriasis was sort of in between. You could see a sliding situation here with a lot of genes involved in keratinocyte and epidermal inflammation or differentiation. So already in the non-lesional psoriasis, there is a beneficial environment for new plaques, as we know, because many of our patients will progress with time. And then, so what does all this mean? We know that there's a difference in the methylation pattern on an epigenetic level. Does this mean anything for the pathology? And Shruti Naik, during her postdoc in Elaine Fuchs' lab at the Rockefeller, did an absolutely mind-blowing experiment to me. Very simple. She treated mice for 6 days with imiquimod to induce psoriasiform inflammation. And then she let the mice rest. And these mice completely go back to normal. You can't see that they have been treated. After 30 or 180 days, so 1 month or 6 months after resolution of these lesions, Shruti just took a punch biopsy and gave them ulcers. And what was very striking was, if you had previously been exposed to inflammation, there was a memory that would very quickly initiate wound healing. And this memory with accelerated wound healing lasted for up to 6 months. This was not dependent on macrophages, dendritic cells, T cells. This was completely stroma cell driven. And looking at the epigenetic, and this is now using ATAC-seq, so the openness availability between histones, there's a huge difference at day 6, as expected, between controls and imiquimod treated skin. Differences persisted for 30 days, 180 days. And here you can just see ATAC-seq data. This is what you get, lots of peaks with available genes. And AIM2, a part of the inflammasome, remained differentially opened in skin that had been exposed to imiquimod a long time ago. And so Shruti did this beautiful paper and model where you really can see here the unwounded skin, and 12 hours post-wounding, the previously inflamed skin is just ready to go, ready to do hyperinflammation. And this reminds a lot of the psoriasis phenotype, of course, with hyperproliferative environments in the skin. So there's an inflammation. Inflammation-induced chromatin states during the inflammation, but after resolution, this inflammation-induced chromatin states will persist and is ready and alert to respond to any new insults. This AIM2 promoter availability was also increased in psoriasis plaques. So it all comes together in the nice round story at this point in a paper 2 years ago, where again, and here transposase accessible chromatin was looked at ATAC-seq in biopsies from normal individuals, psoriasis, lesional or non-lesional skin, and here methylation. Let's see where we are here. Here's openness versus methylation, so you can also see at a correlation. When you have a lot of methylation, the gene is closed. When you have open accessible peaks in ATAC-seq, the gene is open, and you could see the correlation between the expression pattern and the ataxic peaks, and the methylation and the AIM expression. And again, just to summarize what you need to think about looking at epigenetic changes when you look at methylation, when you have a downregulation, hypomethylation, you will have more gene expression. So this is basically where we are right now. There's a fibroblast paper that you will have to read that came out very recently in DID as well. But we have tissue-wide alterations in methylation and gene accessibility during human psoriasis plaques. And in murine models, this has a profound effect on wound, on healing of wounds. And it, as I see it, all this pro-inflammation may lower the threshold in the loop between interactions between T cells and the stroma. And it will be very interesting to look at single-cell resolution linked to transcriptomics and proteomics to understand the long-term disease imprinting in result lesions. So I'm now coming back to what is also very close to my heart, resident T cells, just to see, could epigenetic changes in stroma cells influence the functionality of resident T cells in result psoriasis? I mean, this is the big question for me, but I will just back off a little bit to work done in my lab, and this is only CD8 T cell, because the CD4s are too difficult for us somehow, so we leave them. And we have focused on just looking at functionality of subsets of CD8 TRM cells in healthy skin. And here you can see an old study by now where we defined cytotoxic TRM cells marked by CD49A. These are dependent on IL-15 and TCR ligation to become activated. They respond very quickly to activation and are enriched in vitiligo. On the other hand, CCR6 positive or C positive cells are more prone to IL-17 and are enriched in psoriasis. To understand if this is also reflected on an epigenetic level and how defined these subsets are, Elena Hoffer, who's back here, together with a postdoc from Jan Bryson's lab, Beatrice City, has worked for a number of years now to try to understand how deep the subsetology goes when it comes to resident T cells in the healthy skin. And you can just see here that RNA-seq really defines dermal CD69 positive cells, CD103 positive cells, epidermal CD103 positive cells, always in blue, and epidermal 49A positive cytotoxic cells, always in red. The same pattern was true in ATAC-seq, but it really defined the CD49A positive as a separate entity. And when we look at the open regions in the ATAC-seq, we could see that they spanned over all the parts of the genome, including the exon promoters, etc. And if you look here at coenzyme B, this is just an example, we find more in the 49A positive cells as compared to the 49A negative cells in epidermis, whereas interferon gamma can be found in both. And then the complexity is that there are several peaks, so you might get several results from the same gene. To look at the availability, the promoter availability, we looked some 60 kilobyte upstreams from the start of the gene. And here we could just look at different patterns of recognition, always the dermal, epidermal cells with yellow, green, blue, and red. This is how we talk about them now in our lab. We could look at the first cluster with downregulation of availability in epidermal cells, upregulation, downregulation in 49A, upregulation in 49A. And as expected, we found that the cytotoxic cells were pretty clear, and they had granzyme A, B, perforins, granulysins, CCL4, and interferon gamma. Similarly, 49A were sort of lacking CCL20, IL-17F, IL-23 receptor, CCR6. To our surprise, there was also some shared sites which included IL-17A and F, IL-22, interferon gamma, granzyme B, messy population, a messy mixture of things for one cell. And this explains something that I could never quite explain to myself. When we look at CD49A positive and negative cells in vitiligo, it's very clear, also in healthy skin. 49A negative cells they make IL-17. We never find it in the cytotoxic 49A positive cells. But in psoriasis, it is a bit of a mess. And both subsets can make IL-17 with or without interferon gamma. And here, sort of, I start to understand some sort of plastic, functional plasticity in these subsets. So that was really exciting to me. Coming back to Rachel Clark's beautiful finding, there was a very recent publication from Rachel's lab showing that T cells in the skin, resident T cells, can be derived from central memory circulating cells. And here you can just see the data where you can see it after 3 weeks or 6 weeks in the model that Rachel already explained to you. Central memory cells are very good at providing precursors for tissue resident memory cells. When we look at single cell data, and here in blue we have epidermal CD8 cells, and in red we have the blood cells, CD8 cells again, and the different subsets with 49A positive and negative cells. We see an overlap in the clones between blood and epidermis, both for 49A negative and positive cells. And you can see here that the most prevalent and most expanded clones in the healthy epidermis seems to be quite restricted to the skin. They're not so much shared with the blood. But in between, there's quite a lot of sharing between central memory-like clones and 49A positive clones. So we use this as a way to look at the environmental factors necessary to propagate CD49A positive cells in vitro. And Beatrice and Elena set up an assay where we use Matrigel together with IL-15 and TGF-beta to propagate TRM-like cells. And as measured by their expression of CD103 and 49A, this is quite a nice assay. We get a good population of 49A positive cells with a transcriptome profile that is very much like what we find in healthy skin. Going back to the ATAC-seq data, RUNX 2 and 3 always came up as differentially expressed. And here we see that both RUNX 2 and RUNX 3 transcription factors were necessary for these cells to develop into bona fide TRM cells. So we come with a new idea here on how TRM cells develop. They can both proliferate in situ. I don't talk about that today. Elena will talk about it later during the week, and there are some publications from graft versus host disease that also shows that TRM cells can persist within the skin, but they can also be recruited from the blood, and they need a lot of local environmental factors to develop into one way or the other. So why am I talking about this? Well, I think it's very important to remember that stroma cell alterations probably skew the functionality of generations of local and newly recruited T cells in resolved psoriasis. And there we come back to the longevity of the localized disease memories in psoriasis. We have a dysfunctional pro-inflammatory environment. We can pull in new cells. They will be raised in a slightly dysfunctional environment, and this will just go on and on until we find a good way to break it. So this brings me to the end. Epigenetic inflammation and epigenetic inflammatory wiring of stroma cells sets the stage for T cell-based disease memories. And as I have talked about, we have epigenetic changes that are happening in the inflamed skin. Probably, if the stem cells, in line with Trudy's data, remains poised to psoriasiform inflammation, this will just go on and it will affect TRM cells that are persisting in the skin, but also new cells. So it will probably not be enough to just eradicate T cells in situ. We need to affect the environment, just as Rachel told you. So these are two complementary presentations and views on the same mechanisms and where we go from here. And I just want to finish with a thank you and a few pictures that I found in Tokyo and Kyoto. This is the wonderful people in my lab who very heroically some came with me and some moved on as I moved from Karolinska to Copenhagen. And this is also a result of a wonderful and long-term relationship and collaboration with Jen and Bryson, and our families are now friends after all this endeavors of trying to understand TRM cells, and of course the funding. Thank you.
We have some time for questions.
Hi, this is Mrinal from Michigan University. We see there are epigenetic modification like CpG hypermethylation changes, but do we have any upstream pathway like DNMT3, like DNA methyltransferase changes, like changes in the upstream molecules, not just effect, like what are the causes?
Tissue-wide or in our T cells?
Any specific cell type as well as the tissues, tissue-wide? Yeah,
not that I have found, but I know there are a couple of speakers after the break that might be able to answer this question any better than me.
Okay, thank
you. Well, I got one question, Liv. Great talk and congrats on your paper. One thing that is kind of interesting is that all these T cells, particularly CD8s, have such prominent expression of cytotoxic markers, right? The granzymes.
Yeah.
But yet cell death is really not a feature of psoriasis. What is your take on that?
So they have a lot of granzymes, but not a lot of perforin in psoriasis when we look for it. And there's also an interesting expression of granzymes that together with IL-17 seems to affect keratinocytes. So we had a small observation on that. But I don't think the cytotoxic machinery is really working as it should in psoriasis.
Okay.
So that would be my explanation to that.
Any idea why?
No. They don't know. No, there's so many things I don't know. But I guess it's environmentally skewed. And this is, so I've been trying to, because I was always puzzled by this thing that CD49A positive cells are so cytotoxic, and yet they can express IL-17 in psoriasis, and there they don't have perforins. So why is that? And we've been trying to set up cultures to look at functional plasticity. It is so hard. Once they are into the 49A program, it's difficult for us in vitro to get them to go into more psoriasiform phenotypes.
Yeah.
So I think there's a lot to learn from the stroma.
Okay. We've got a question. Chris?
Chris Griffiths, London, Manchester. Yeah, this is a complex area, Liv, and I think you did a great job of trying to explaining this for us. You mentioned on 2 or 3 occasions about the role of fibroblasts. And I remember back, because I can remember this far back in 1986, there was an article in, I think it was in The Lancet actually, by Colomb and from Paris, where they used living skin equivalents and transplant and used psoriasis fibroblasts and normal fibroblasts, and showed that if you put the psoriasis fibroblasts into the normal skin model, you got psoriasiform proliferation of the keratinocytes. And it's always intrigued me as to the potential role of fibroblasts in psoriasis. And I think now we're starting to see some evidence that they are a sort of partly paid up member of this immune response and actually are producing growth factors which are driving the epidermal changes. I just wonder what your views are on that.
I completely agree. And I was also reading that paper. I couldn't fit it into my story in a good way, so I sort of didn't. Sorry if any of the authors are here. But it was, I think it's definitely an interaction. And when we look at, so when we work a lot with fibroblasts and keratinocytes in parallel, when we try to activate them with different cytokines, fibroblasts often respond much stronger. But I have a very niche interest in the epidermal compartment, so we sort of, we don't focus on it, but absolutely, they're very interesting. And I hope there will be more papers coming out over the next few years.
Great. Thank you, Liv.